Did you all get a message?
Yes.
Okay.
You are
great.
recording.
We are recording. Very good technology. So I'm Dorothy Dillard. I am the core lead for the Community Engagement and Dissemination Core of DSU's iHer Center, which is an NIH funded project. And iHer stands for the Interdisciplinary Health Equity Research Center. And we aim to increase the connections between our researchers at DSU and our community partners. We had the good fortune about a year ago to be awarded funding from the Delaware Health Science Alliance to conduct a small project with the Delaware Breast Cancer Coalition. DBCC has an incredible messenger program called the Chanpyon program that trains and educates women in the community to teach their community members about breast cancer and breast cancer prevention. They have adapted the program for the Haitian Creole population. We are very interested in partnering with DBCC to take a look at how that program's working, provide a small scale evaluation so they had some data to be able to look at their work and use that evaluation, those evaluation findings for seeking funding, marketing what they're doing. So that was their benefit of participating. And then we also have a component in the project that brought our researchers from DSU and from BB Healthcare to meet with the champions, the Haitian Creole women who had been trained and working as champions in their communities, to learn more about the research around breast cancer that's happening here in Delaware. So I had the good fortune of working with Scott Siegel at the Helen Graham Cancer Center at Christiana Care, two of my colleagues at DSU, and Dr. Dixon Whitmere at BB to coordinate and present on what they're doing. Scott served as a great thought leader for me. So we'll go around quickly now and just do quick introductions of everyone. And then we'll begin with some questions for the researchers who presented to the champions. So Scott.
Hi, thanks for including me. My name is Scott Siegel. I am the Director of Cancer Control and Population Sciences for the Helen F. Graham Cancer Center and Research Institute at Christiana Care. We focus mostly on addressing breast cancer disparities through the application of behavioral and population sciences to address the disparities statewide.
Erin?
Hi, I'm Erin Perchiniak. I'm an associate professor in the biology department at Delaware State University. And my lab focuses on a specific gene that's normal job is to kind of control growth in cells. And there's different variations of this gene in certain populations, and we're just studying to see if there's any differences between this gene and different populations, specifically African-American lineage.
Carl?
Good morning, everyone. My name is Carl Miletti. I'm an associate professor in the Department of Biological Sciences at Delaware State University. My work mainly is the analysis of phenotypes of cancer cell lines, of triple negative breast cancer cell lines derived from African American women and European American women.
And last but not least, Diana.
Hi, Diana Dixon Whitmer. I'm the medical director of the breast program at BB Healthcare. I've been involved in public health for more than actually 45 years, was an advisor to the Division of Public Health in Delaware and had worked with public health resources in New York before coming to Delaware. And I'm a breast surgical oncologist. I'm very interested and involved in clinical trials. And as you know, accrual to clinical trials among minority populations is poor, and that's a problem. And I've been devoted a lot of my time to seeing what can be done to improve accrual. So this was an effort on my part to try to understand some of the barriers to accrual into clinical trials.
Great. And I owe a deep-seated gratitude to all of you for your participation throughout this project. I think we'd all agree that it's been interesting, and we've learned a lot, and it's been fun. I've had fun doing it. It's probably in my 40 years of doing community engaged research, one of the funnest programs I've worked on. But it's rare that we have folks working in labs doing pretty technical, you know, scientific work, big words that I don't even understand, meet with uh, our peer educators, our credible messengers, particularly a group that English is not their first language, and some of them don't even speak English. So that added an extra element of fascination to the project. But I want to start with why you agreed to do it. What did you want to get out of participating? Everybody's really busy. We have to pick and choose what we do. So why did you agree? Why did you agree to do this? And we'll start with Erin on that.
So I think I was trying to think about back to when we started talking about this, and so I happened to be fortunate enough to be part of sort of a big grant that's on campus, and I had written a small pilot for it, and part of it was you should be involved more in community engagement, and I didn't really even think I understood what that meant. So we were kind of paired up with Dorothy or a few of her colleagues to kind of chat about how the research that we do in our labs could affect your local community or broader community. And so I think I just had never been asked to kind of consider that much before. And so I was excited to learn about what that would mean. I've lived in Delaware most of my life, so I certainly care about, cancer in general in Delaware, certainly breast cancer in Delaware, and understanding different, like Dr. Whitmer said, barriers in the community that help, getting people in for access to care and things like that. So I generally enjoy learning about that topic, but I think it was more the connections on the individual level on campus where after chatting with Dorothy, I was like, oh, I'd love to do that. But, it had never really come on my horizon before. So that's why I jumped at it when you asked.
And Carl, why did you agree to participate?
I'm gonna say that it was mostly because I was curious. This is my very first time that I am in contact with such a group. My work has been always in the lab, and like you say, we go there, we do 20 different experiments, and see what happens. But this interaction with the champions was really really interesting and growing for me as a researcher. I guess we'll have a chance to talk a little more about that later. Uh, but like I said, was more the curiosity. How would this go, and what would they think about my work once I explain?
Yeah. And Diana?
I think it's been a part of my mission in going into medicine to work with public health. The thing that took me to medical school had nothing to do with surgery. I did a project in Harlem studying what percentage of individuals who'd had amputations and had prostheses were able to walk a year later on their prostheses. It was 8%, by the way, in 1968. And in doing that project, going door to door in East Harlem, I learned 11 people can have to live in one room and the bathroom's down the hall. And there was this huge tuberculosis epidemic in New York at the time. And it was such a surprise to healthcare people because it was sensitive to an antibiotic that was inexpensive. And it was obvious to me, this isn't about the antibiotic and the bug. This is about the social circumstances under which this population is living. So I only went to medical school to try to address that kind of problem. And then I discovered surgery and fell in love. And I have thoroughly enjoyed being a surgeon. But I try as often as possible to bring myself back to the original mission and continue not just to work on this person that's right in front of me and with that person, but to work with a larger community. So I thank you for the opportunity.
And that's a really important connection there with that we can discover things in the lab, we can learn new ways to do better surgery, all those kinds of things. But if it's not translated, so that's what this is
Hmm.
really about, right? In a way that is practical and doable for the folks who need it, then it kind of misses the mark, right? So I think that that's what we're all have a you know sense of curiosity about as well, and a desire to address that. Um, so I came to each of you and asked you to do a presentation about your research for a group of Haitian Creole women who the Delaware Breast Cancer Coalition had trained to be credible messengers in their communities, um, teaching their peers, their community members, their family members about breast cancer, particularly breast cancer prevention, and really promoting annual mammograms. So that's kind of the pitch that I gave to you. Um, in thinking about that, how did you prepare to do this presentation? How is it different? What did you think about? And again, I'm going to start with Erin on that.
So I asked a lot of questions to start. So I certainly talked with Dorothy a little bit about who the audience might be and how many people and sort of what background they might have. Usually that's what I do when I teach any classes. I'm thinking about who's sitting there and what, where they're kind of coming in. So she said, I think start from the scratch, with what you're going to talk about. And then it took me a little while to think about, I certainly didn't want to talk about the specific gene that I work on that much because that seemed a little too in the weeds. So I actually just talked about how, changes in your cells, and we did talk a little bit about DNA, but how changes can lead to things like cancer. I tried to use as many sort of pictures as I could of things, and really it just turned into a conversation. So I did bring slides, but we really wound up talking more than anything else. But yeah, I sort of just started with what would I want. I was trying to think, what would I want to know about what I do, in regular terms, if I didn't really, know a ton about science. So that's sort of where I started.
And Carl, how did you prepare?
In my case, was easier because I just went to Erin. I said, Erin, what did you do? Let me see your presentation, and it was kind of the same format that I used for them. But then I tried to continue where she ended, and then I tried to just go a little more into what triple negative breast cancer was, and why I wanted and why I was doing that type of research in my lab. How could that hopefully inform us of uh, targets that we can use to treat patients of this type of cancer? Uh, but I also thought about the audience, as Erin just said, uh, and I was really excited with the questions that they were asking. Even more excited that I was able to answer them in a way that they could understand, and not too technical, like you said at the beginning. So that's how I prepare.
So, and Diana, how about you? How did you prepare?
Inadequately. I went to my collection of presentations and looked through the National Cancer Institute sets for ones that explained the basics of clinical trials. And then I tried to modify them to be in more understandable language. But what was totally inadequate about that preparation was that I did not understand that my audience did not speak English at all. And had I understood that, I would have used one of the technologies to translate into Haitian Creole the slides, because they just had some things up that meant nothing to them. And so next time I'll be smarter.
And we all learn through this process, and I'll be clear also. I don't think that I realized that the English was not, that there weren't more who um, had a little bit better understanding of English. We had an incredible translator. Just it
Mm-hmm.
still amazes me how well Margaret translated. Um, but
But
I
hey,
made
this is part of what we've all learned from this, is that population, here's another one of the barriers. They don't speak English, read English. They are completely cut off from routes of communication that we think is useful.
Right. And it also highlights, I'll come back to that because that's a really important point as well. But it also highlights our assumptions. When I was told that they don't speak English, I interpret that to mean, when I'm told that a group of Latinx don't speak English, that there's usually, some understanding of English. And so it may take a little longer, but it's not a complete break from one language to another. So that also was, a lesson learned for me. I do want to make this connection that's coming up now, and really highlight it, because it is one of the things that we learned. It is around the language barrier for Haitian Creole, that is one different than what we see with Latinx, for instance. But we did a community conversation for another project with the Haitian population in Seaford. And when we asked them questions about health literacy, understanding what their doctor said, it was really startling and concerning that that same language difference that we experienced, they're experiencing in the doctor's office. And that, there's a telephone translator, that translation that they can use, that's all that they're given, and that's woefully inadequate. So I think we're all putting that on our radar to make sure that we pay more attention to. um, I also want to be clear on the fact that I did not set up these presentations. I did not schedule them in any order, and the process just worked out so beautifully. Aaron, you started out talking about biology in general and what
Mm-hmm.
happens to cells when, as you said, they misbehave. And then Carl knew that and was able to build on it, and really share much more about what happens in the lab, and had slides of the petri dishes. Carl, can you share just a little bit more about, listeners can get an idea of the level of scientific information that you provided?
Yes, I was trying for them to imagine, to visualize how we study those kinds of cells in the lab. Because I say it because I work with that all the time, but I don't know if people can really imagine. How do you get cells in the first place, and then work with them in your lab? So that's when I came up with the idea of showing some of the instruments slash vessels where we put the cells, where we work with them, and that's why I show some of the plates. Uh, and I believe I explained a little bit about what are we looking at when we're looking at cells in a plate. How can we compare uh, the aggressiveness of one cancer cell line with another, whether they come from different people, from different ethnicities, or the same. And then we look at how they behave based on, as Erin was mentioning, different genes. And I don't know if I use the word genes, hopefully not. Uh, and how these cells then start behaving based on that. Yes.
And some of the questions that came up, one of the big questions was, how do you get the cells? Which set us up perfectly. You explained that in a limit, just in an introductory way.
Mm-hmm.
And it set us up perfectly for Diana's presentation on
Mm-hmm.
clinical research and clinical trials. And so, Diana, do you want to talk a little bit more about kind of the level of information that you provided to this group, and what level you thought what they understood the most to help people who are listening and think about how you introduce some of these more technical things.
You helped a great deal, Dr. Dillard, because the concept that was key to understanding how clinical trials answer questions is randomization. And you literally did a physical demonstration of randomization in the room. And that was perfect because that visualization of what randomization is was, I think, the most important message, along with the message that you are safe if you are treated in an NCI sponsored clinical trial, that you are not at risk of having a worse outcome than had you been treated by your doctor doing whatever he or she thought was best outside of a trial.
And I think one of the other points that you made that was so important was that there are different kinds of clinical trials. And so most likely the kind of clinical trial that this group or their, friends or family, community members would most likely be asked to participate in would be after the research and trials to ensure that there's some level of acceptable safety with whatever intervention or drug medication. So I thought that was really important. What was this experience like for you, Diana?
Enlightening. It was really very enlightening on several levels. One, I knew that there was a large Haitian population in Western Sussex County. I'd known that from work that Scott has done in the past, work that Jennifer Sims Murtada at Christiana in Translational Research and I had done on triple negative breast cancer. But I did not know really how very isolated they are. The language issue, I don't remember whether the discussion was during that visit or offline, but understanding that many of them cannot apply for Medicaid because they work as cash under the table laborers in people's homes or elsewhere, and so they cannot declare their income, and so they have no visible income, and the barriers to getting Medicaid coverage are now more understandable to me. Also, it was enlightening in that there were people from Delaware Breast Cancer Coalition there, and I've worked with the coalition since I was actually one of the founders with Maureen Lauterbach, and I thought DBCC was educating people about clinical trials. The peer mentor group was a big part of its mission, was to educate people about clinical trials. But the Delaware Breast Cancer Coalition people knew nothing, and apparently that is not anymore happening. So that's an opportunity identified for those of us involved with DBCC to reinvigorate the education of volunteer survivors in the peer mentor group to be teaching about the safety and importance of clinical trials.
Yep, that's one of the things that resonated with me as well, is that there's just such a lack of understanding of that, and it's in the community, you know, everyday people, and that's who we need involved, right? Um, and really thinking about more education and promotion of that. So, Erin, how was the experience for you?
Yeah, it was really eye-opening. Similar takeaways that, I certainly, know that there's barriers that exist in populations and, kind of conceptually understood that, but to see it in the room was a very different experience for me. And I always teach in biology classes that even if, you don't wind up in healthcare or being a researcher or anything like that, that you're going to be one of the more educated people in your families about things that, are important for your, healthcare. And so you're going to be your patient advocate for your family. And, so we always chat about that in class, but I was thinking about it when I was chatting with the women, obviously through a translator, but, that you only get so many minutes with a physician in a room. And if you don't speak the language at all, like not even understanding any of it, you're only ever going to get the basics. You're never going to get those other questions asked that I might fire away and be like, oh, asking. So it's not even just education level, but also the language barrier. It's just it blew my mind, like how much that puts you at a disadvantage when you're sitting with a physician,
Mhm.
and that, it's not even, you don't know what question to ask, you don't even understand what they're telling you. And so it just really hit home that how difficult that would be, in cancer, so complicated that if, you're only hearing these drips and drabs through a translator, and a wonderful translator, it just makes it seem so difficult. So that was my takeaway.
And Carl, your experience?
My experience, I can describe it in a couple of words. It was novel for me. It was exciting doing this for the first time. Challenging at the same time because I needed to transmit some information that I was not used to do in those words. Uh, and maturing. It's really helped me take a look at my work at my work uh, through a different set of eyes, and hopefully in the future, uh, and that's something that comes to my mind all the time. I would be able to work in something even closer to them, maybe going from cells to tissues, and so we can get closer and closer to the person who hopefully benefits from your work.
And that's what the community engaged research is about, is how to do that. And I think that on the intervention and the program, and we really have included the lived experience, included those who are receiving those interventions, but still need to do work across the um, translational process, and making sure that we know who what those barriers are with the people that we're working with. and
Mhm.
and to your point, Carl, also thinking about how to put put what you've done for, how many years, how many decades you've been working in this, and it's a second language to you, to back up and think about, how do I put this in terms that
Mhm.
anybody can understand? So, yeah. Anything surprise you besides the language piece of it? I'll just open that up if anybody wants to share other surprising pieces.
I had an interesting, so we had a very specific conversation in my week that I was there, and one of the women asked a question about the 40 year olds, as the marker for getting a mammogram. And so this really good conversation happened where somebody was commenting that they couldn't get their friend to go to an appointment for a mammogram because they were 35 and they were having breast pain and, other issues that would, trigger you to want to get a mammogram and qualify you to get one. but they couldn't convince the woman to go because she's like, but I'm not 40. So to me, it was surprising that like some information is getting, like the 40 message got in, but then there was all these other nuances to it that are, really complicated. But it was another limitation. It wasn't that the woman wasn't planning on going for her mammogram. It's just that in her mind, it was 40. And why would she ever think to go earlier? Or if there was a family issue or things like that. So we had this really good conversation about that. But the champions were saying that they have their own barriers where they can't convince people because there's sort of some information there, but not the full story. And so that was really a good part of our conversation.
So Carl, back to you. What was the most satisfying part of participating in this
Most
project?
satisfying part, I'm going to say it was that exchange of questions and answers between me and the champions through the translator. Um, I was very happy that I was able to inform them a little bit of you know the topic, and I do remember that they did not realize that we're more than one type of breast cancer, uh, and that some are more aggressive, some show up in younger women, etc. Uh, and I have to say that I was very excited to be able to give that little piece of information to them so they can keep an eye on their population and make sure that early symptoms are caught on time.
Diana, what was the most satisfying part for you?
They seemed to, after a while, not at the beginning, but they seemed to really be very happy to know that uh, that doctors cared about them. They seemed to be happy that they were having a direct, through an interpreter, but direct dialogue with somebody who was potentially the doctor of one of their friends, neighbors, or relatives. And I felt really good that they uh, appreciated having direct contact with someone that I think they think of as being part of a group that they really can't talk to. I'm not expressing this very well, but after we were halfway through, and especially after you did the physical demonstration of randomization, I felt that they were very engaged with the discussion then, and that I had a rapport with many of them that hopefully will translate into being something that reduces one of the barriers, the fear of the doctor's part, if they do need to come for care.
I picked up on that too, and every one of the sessions, it always took a little while to kind of get warmed up, and then they were fully. I mean, it was like from are they paying attention to okay, now we're fully engaged.
Mhm.
That was a very satisfying piece as well, and clear appreciation for being there, and I think realization on their end that researchers and doctors, they're human beings. So Erin, how about you? What was the most satisfying part for you?
Similar. I think it was that it wound up being very conversational. So I think at some point they sort of settled, and so did I, that, in on like this would, just a bunch of women in a room talking about something, and they knew things I didn't know, and I knew things that they didn't know. And so then we could share back and forth. And I do feel like about halfway through, similar to everybody else, that, I feel like they felt like, oh, she's not, I don't know. We felt more like we had more in common. And so I thought that was really helpful. And I found that rewarding, that it just seemed like a conversation where I wasn't there to like teach them something necessarily. It was just a conversation.
Were there any disappointments?
Only in myself. I, like Dr. Whitmer, I wish that I had put my slides either all pictures and no words, or just had them translated, because you can use programs to do that. So I'm sure I could have, or I could have asked Margaret to help me. So I wish that I had come more prepared for that. So just disappointment in myself, certainly not in the ladies.
Well, and that's a lesson I learned also. I mean, that's maybe one of the good things that AI can help us with, right?
Mhm.
For all the other scary things. But Carl, any disappointments for you?
No disappointments. It was a phenomenal experience. Uh, I hope I can participate in the future again, if it's needed. And I just want to piggyback on something that you said, and Erin also mentioned. And it's for them to see that we are people same as they are, and we're not much different from them. And I believe I share with them the number of relatives that are in my family that have had cancers, all kind of different cancers, all coming from my dad's side, and that we also have to deal with something like that, whether as a caretaker or patient, or and I hope they took that message. Uh, we we are very similar to them. We go through the same struggles, uh, and definitely there was no disappointments whatsoever in this experience.
In Carl's session, we also had the privilege and the honor of his wife attending, and so she jumped in on a couple of occasions to share, what it was like as a family member, and somebody that that also experiences cancer and dealing with it on a
Mm.
human individual level. So, Diana, was it worth your time?
Oh, totally.
No. So I'm going to go back and kind of wrap up this description and your feedback on the research presentations and the experience by asking you to kind of sum it up in 3 to 5 words. I think we already heard from Carl. We heard novel, exciting, and challenging. So any other kind of
Enlightening.
enlightening?
Informative.
Maturing for me.
So, um, so those of us who are centered in community engaged research and work to increase the community involvement and research across that scientific spectrum from the lab to the patient and to the community, frequently find some resistance from our bench or lab research colleagues in doing community engagement, thinking about community engaged research, how to involve the individuals who may be affected by whatever you're researching. They often don't see that they have a community, or feel that um, they feel that their research is separate and kind of needs to happen without regular people involved in it. And they don't really understand the importance of community engagement. So after this experience, what would you say to your colleagues to help them better understand that the importance of and need to participate in community engagement activities? That was a whole lot. Did you all follow? Carl, I'm going to start with you as
Okay.
a
I,
spending most of your time in the lab.
I would tell my colleagues, make yourself available. Participate whenever you have a chance. Participate in this type of community engagement activities. Uh, in my case, I did not only have the opportunity to inform them of what I do, but they also, knowingly or not, they inform me of whether my work is going in the right direction. Maybe I can fine tune it. Maybe I can modify something, and that those modifications hopefully will go towards helping them with their needs and expectations of what the science behind all this is doing. So it was an amazing experience, and I thank you for this opportunity, uh, because I definitely will do it again in a heart.
Well, and you've been asked to do a presentation at the Hispanic Heritage
Hmm.
event later this month. So,
Yes,
uh,
I'll be there.
and you can now speak and um, the- to the Latinx population.
Hmm.
So,
Absolutely.
Diana, what would you say to your colleagues?
I, to my colleagues who also participated, but who are doing the hardest work, which is the bench science part of this, if we can't get to groups like those in Western Sussex, if we can't get them to have genetic testing and find out what are the differences in different populations in their genetic makeup, if we can't get them to participate so that tumor is available for research, then your ability to develop new treatments that are broadly applicable is so limited. Maybe right now, the participation of Blacks in clinical trials, including the trials where it's collection of tissue, that's a big part of it, is so low still that the results are not always applicable to that population. And so they get doubly hampered. They aren't included in the clinical trial, so they don't have the chance to be in a treatment arm that turns out to be an advancement. And because they're not included, their tissue is not part of the tissue bank, and you all are not able to be studying the tumors they have and their normal tissue. And so the results of the trials are not benefiting them. So I think it's wonderful that you guys were doing this and hope you keep doing it on into the future and inspire your colleagues to do the same.
And Erin, what would you say to your colleagues?
I would say, first of all, I think sometimes we just don't know that, like, this was the first time this ever was an opportunity for me. So I think sometimes we just don't know about these things. The other thing I would say is that scientists are trained to only say that things are true after a lot of data. And so we get very nervous talking to like patient populations because we are, you don't want to share something that, doesn't either make sense or that is not the full story. So I think we're just trained to be so careful about saying that things are true or whatever. And so I would tell them to not be so worried. There's definitely things that you can offer, even though we're not clinical people. And that, that was my reservations. Like, I don't want to tell anybody that, they were asking questions about mammograms and things like that. And that's not my wheelhouse necessarily, but it was okay. We just had a conversation, and then, we can get them the right information and all that good stuff. So I would just tell them not to be afraid to do it because, I learned a lot. It was a really great conversation. I would love to do it again. And, there's a lot to learn from it, and that we do have things that we can offer, groups in the state as well, and hopefully be helpful in some capacity, but not to be afraid of it so much.
To be curious, right? The
Right.
same thing that brought you into the lab can bring you into the community conversation.
Yeah.
So, yeah, yeah. Scott, I want to give you an opportunity as someone who's also doing this work and has been involved in the kind of creation of this project. If you had any questions, thinking about, encouraging others, and bench researchers, lab researchers, clinical researchers, to have this community involvement.
Well, I was interested to hear everything everybody said today, and one comment that Diana made resonated with me because I do work with a bench and translational scientist at our center, Jennifer Sims Mortada. And as we started looking into just the representation of tissue in these preclinical studies, it's still abysmal. Even today, it's not as if we've improved things in the last 20 years. The focus on triple negative in our state, given the incidence rates, really the disparity emerged between Black and white women going back to the 80s. And we think there's two things driving that. One of which was we developed better targeted treatments for hormone receptor positive breast cancers and not so much for triple negative breast cancers. And when you go back and you look at those studies on tamoxifen, the first targeted treatment for estrogen receptor positive breast cancers, it was 97, 98% of the participants were white. You would think we would have learned that lesson, but even in studies today, published in the last year or two on triple negative breast cancer, which Black women have twice the incidence, we still see overwhelmingly that those samples are white women or of European ancestry. So the only way I think we make progress in improving treatment is doing the work that you've been doing, Dorothy, and everybody else, is building trust, having a conversation, dealing with the language limitations, of course, but we need the community to be in the trials. We also need the community's perspective on what those trials should look like. And the bench scientists will not be successful in their pursuits if that doesn't happen. We'll just keep studying and improving outcomes for a segment of the population, that has been advantaged all along. So I think this is wonderful. It's hard work. We need to do a lot more of it.
And I will say, I think that that's a great way to wrap things up around the necessity and the importance of this type of work, because I will say, if Carl went back to this group of women and asked them to participate in a clinical trial, if Diana did, they would jump up. If Aaron did, they would jump up and say, yes, I'm all in. They were asking, how do I, or someone I know who's going through cancer, how do we provide get involved in a clinical trial or provide our tissue or whatever. So it's amazing how, even as limited as it was, that interaction, just that personal interaction and interest and respect, just opens up all kinds of doors.
Thank you.
Again, any last comments? So again, many thanks. I was kind of operating in the dark, throwing spaghetti at the wall, and I am very grateful for how well this went, and how beneficial it's been to you all and to the champions. And I think we've got just a great model to see what we can do with sharing it with others as a way to increase that connection from one end of that scientific spectrum to the other. So thank you again. Appreciate everybody's.
Thank you.
Thank you. Thank you, Dorothy. Thank you all.
So I'm going to try to stop.